Madhushankar Edirialaji. He obtained his basic degree, BSc in Radiography, from the Faculty of Allied Health Sciences, University of Peradeniya, and he has obtained his PhD from Tianjin Medical University, China. He is currently working as a senior lecturer at the Department of Radiography and Radiotherapy, Faculty of Allied Health Sciences, General Sir John Kothalavala, Defense University. I'm so excited and proud to welcome Dr. Vasana, as he is one of my batchmates and he is one of the first alumni of Department of Radiography, Radiotherapy, Faculty of Allied Health Sciences, University of Peradeniya. I truly express my gratitude to you, Dr. Vasana, for accepting our invitation today. So before we start the session, this is a kind reminder to all the participants today. Please mute your microphones to avoid background noises. So for further ado, it is over to you, Dr. Vasana. Thank you very much.
Thank you, Bimali. Can you see my screen? Hello, Dr. Bimali, can you see my screen?
Yes, Vasana.
Okay. Thank you very much for kind introduction, actually. Can you see my face here? I have switch on my camera also.
Yes, of course.
Okay, thanks. Okay, actually, thank you very much again for your kind introduction here. Actually, you all are welcome to my session. Today, actually, I had in my mind this topic long time ago to conduct for youngsters and also radiographers or colleagues in the field. And I started making this presentation a long time ago because when I was studying in China, I really wanted to share my knowledge, which I gathered in outside or otherwise in China when I was doing my PhD. So I will try to my best to deliver my knowledge as much as I can, but I'm not sure whether I can conclude everything within this hour, but I will quickly go through all these techniques available to do research in the aspect of MRI especially, right? So here I have taken 3 logos on top of my my presentation. The very first one, when you introduced me, I went directly back to 2006, from 2006 to 2011. I was in Peradeniya. We have great memories there. Bimali knows everything, and everyone knows, especially my colleagues. And in 2011, we passed out as radiographers. And then I served there for 1.5 years, as I remember, and then I left that place, and then I joined KDU and started KDU radiography program, BSc radiography program, as the second institution in Sri Lanka, which produces radiographers to the nation. And I proudly announce it. And then after 2.5 years, served there to produce roughly about 4 batches, and then I left the country. And I left the country in 2015, and I came back in 2020 after 5 years of hard work. And then I came back and started working again as a senior lecturer in General Sir John Kothalavala Defense University. So throughout last 5 years, I have learned a lot about neuroimaging, because actually that nice story. This is extremely accidental kind of thing. I mean, I never expected to do work or higher studies in the field of neuroimaging, but actually I expected to do my PhD in nuclear imaging. Finally, the person who expected as a supervisor is unable to speak English, so that's why the problem. I changed my track, and finally I realized I have chosen the best field that is actually that is unacceptable. I cannot accept. I didn't accept it because the reason is actually I had even my proposal there, but finally I didn't accept this one because unfortunately or fortunately, I got this area as my field. Uh, the first one I want to mention, the introducing MRI-based neuroimaging research is very important because this area is largely uncovered for radiographers. That's the main thing. This is largely uncovered for radiographers because a little bit hidden kind of thing, because the radiographers are usually what they use produce radiographs. They don't do anything beyond that. So that's the problem. So when we search or when we do research or literature, we found that some areas we can address, we can actually go through, and we can do research, good research, based on MRI. So that's why I had a special interest of going through this aspect and then gathered some knowledge, right? And then my PhD is based on completely MRI based neuroimage, right? So I have added a small running title here in the bottom part, removing barriers for research in radiographers. I'm going to introduce some new techniques which can address, especially without doing any repetitions. I have seen many people for the undergraduate work, they are always trying to do some repetitions or the same thing is again and again. So that is not useful for the society or any community. If you do spend time, if you do some hard work, it has to be very useful for the society. That's my concern. So I'm coming into the next slide now. uh, I have few objectives here to make you aware about current contribution by radiographers towards research and innovations, and to describe most common research method used in MRI-based neuron. This is the main part of the lecture. And then I have a list of toolkits, available toolkits used in neuroimaging data analysis, right? And then finally, I have a few suggestions to enhance professional recognition. These are my major parts of my lecture, right? And if you look at the contribution towards neuroimaging research, I have some. I really wanted to find how the people have contributed this field of research, especially when I go through the papers. I have made a search in 2020, and the majority of papers are contributed by engineers, especially biomedical engineers. That's the major thing that we can found. And then in addition, we have clinicians are coming as the second, and then medical physicists and radiographers are in the fourth place. Means that they have very less contribution for neuroimaging research. The reason might be sometimes now gradually introducing, we have very limited number of people who are doing postgraduate research and obtaining PhDs and MPhils and masters. That might be the reason to reduce the number of papers. But knowledge-wise, you can do not keep any hesitation. You can do any kind of, you can reach high level of research. I have the evidences, and then you can reach that level definitely. And again, I search which modalities are contributed for neuroimaging research. When I go through in 2022, I got into PubMed search, I found MRI is the leading technology or leading modality which governed all this neuroimaging research. Majority is governed by. Okay, and then in addition we have CT, PET, and X-ray. Those are radiation based. That might be the reason. Radiation might be the major problem of not using this in more frequently in imaging settings or any hospital or any health society, right? Health setting, right? The MRI has given some, the other reason is MRI is highly advanced in technique. Every day it's advancing. People are writing papers, people have new findings, they are searching for new areas. These are the very important aspects, and we should be always keep in touch with these findings, and we should try to go through these findings and produce new papers, right? So then only you can produce new research with high novelty. So that's what we want. That's why I'm always touching with these findings, these novel techniques, and I'm always searching for those techniques. And then finally, I want to introduce to my youngsters. That's how I am doing, right? And then I have a very recent example for radiographers' contribution to research. This is a little unfortunate. I mean, the recent example is the KDU IRC abstract submission here. I have given a summary of abstract submission. The radiographers' contribution is very low. It's a 9% of abstract submission as a whole. I mean, the majority is given by, the contribution is given by nursing. There are some reasons. I mean, there are more institutions in country compared with radiographers producing institutions. We have so many nurses producing institution. They are always doing. When we have more number of people, more number of heads or students, they are more tend to produce radiographs. Abstracts and submit for the publishing platforms like KDIRC, which is annual one, which is going to be held very soon. And here you can see we should try to find methods to increase the number of this abstract submission, right? This is one aspect. And I have understood, I have found some barriers in doing research in Sri Lanka. Actually, I started making notes of these facts when I start PhD, because I had a habit when I start working in the mornings, if something is coming into my mind, I just make a note. So I think one day I can deliver this message to my youngsters. So that is how I learned there when I was learning in China. So I have no any fear to express this. There is a huge gap of language. I mean, using, people are very reluctant to do read or write or listen English. That's the main problem in our category, especially those who are working in settings. They don't like to speak English, and they don't want to increase the skills in speaking or writing or listening. So that's why that's the major problem here I'm looking at, right? And then funding and facilities. There are major the funding sources in Sri Lanka. One is National Research Council. The other one is National Science Foundation. We call it NRC and NSF. And in addition, we have the faculty related or otherwise university related funding sources, right? And we can write proposals and we can ask funds. And there is actually, there are if you have good proposals, definitely those proposals will be funded. So there are some commitments. We have to do some hard work. Then only we can earn money, or otherwise we can get funds to conduct our research, right? And then in addition, I have seen that facilities are very low. I mean, as an example, if you are really want to do this research, neuroimaging research, you should have some specific facilities, right? Facilities like, the computers. These are highly computer, we need computer facilities, right? Not just the computer facilities. I mean, we need high-end computers to high-end computers, or high RAM or high performing computers for our the data analysis. For such kind of cases, we need to have a good computer. We don't have that facility. That's one problem. Okay. And nowadays, actually, we are going to establish one lab in our premises in KDU to facilitate all those the neuroimaging research, right? And then lack of supervisors is one other aspect, barriers in doing research in Sri Lanka. I have my experience. I was going around the country to find out one professor or one supervisor for my work, or otherwise for my research. And then I was finally fed up with finding a supervisor, and then finally I decided not to find a supervisor from Sri Lanka, but to find someone from outside. So then I decided to go abroad and find someone from the other country. So that is the problem. Actually, we had a problem of I think someone is disturbing me. So the lack of supervisors is one aspect. The other one is poor motivation. So there must be a motivation to do research. One is, right? And I don't see that kind of motivation that should be coming from actually, as an example, if you are doing research as a radiographer in working hospital, that motivation should come in from the higher levels. I mean, hierarchy. There must be some at least some, like incentives, like, it has to be affected for your promotions, like things. And then those are the motivations should be coming from the hierarchy, right? Or administration. And the lack of institution support is very prominent. I mean, there are no supports at all. I mean, very only for academics can get some support. And the limits and monopoly means, I can give one example. I heard when I come back to Sri Lanka, one of our group of students has started a research in Lady Ridgeway Hospital, and then some radiologists has made some obstructions, because they have told that okay, this belongs to us. This is not belongs to radiographers. Therefore, you cannot conduct this kind of research. No one can make any barriers or boundaries for research. If I want to do research in our language, I can do research. So no one can make barriers. There are no limits. If you want, if you are really interested, you can definitely perform. That it doesn't matter, right? And then ethical concerns, I can say you that ethical concern is very important, because if you are using some vulnerable kind of the groups, as an example, the pregnant people or sometimes animals. So there are some very strict guidelines, very strict the ethical concerns in Sri Lanka, right? So we have to face those things as barriers, and then we have to find some solutions. And then the other thing is personal, family, or sometimes, once you pass out from university, there are no motivation from the family at all, because they are always sometimes even for me, actually, I got some influence from my family to go to hospital and earn money. But I personally took a decision not to become a professional radiographer there. But first, I have to do some academic work, and then at the same time, we can gather those knowledge by working at university hospital. That's how I was thinking. I think I have taken a good decision at that time to become academic, and now today I have been able to deliver my knowledge or share my knowledge with you guys, especially youngsters in the country. And common faults of radiographers, common faults of young researchers, I have identified quantity over quality means always people are trying to produce number of papers. That's totally wrong. I mean, if you are interested in research, you should try to produce quality papers. Not if you can produce only one research or one paper for your within your lifetime, it doesn't matter. You can produce one. It doesn't matter. But if you are looking for quantities, I mean, it means that the quality might be drastically reduced. The reason, if you are targeting the number of papers, okay, I have published this number of papers, you can say. But the problem, if a researcher look at your profile, if those researchers are not useful, those will not be cited by other researchers, which means low citing work, means that low quality work, right? And then the other one is no supervisors involved. Sometimes some researchers conducting without supervision. This is one aspect. Without supervision, you should not do any research. The reason is that, even me as an example, I mean, still I am doing research, I'm conducting research, but even after PhD, I am working under supervision. I mean, if I have any problem, then I'm consulting my professor, and then I ask questions, and then I get the clarifications, right? And then national and international relevance. This is very important, because when you start a research in the country, usually the countries like us consider the national relevance, especially national relevance. Then only they provide funds for or support for the study. So we have to look for national relevance, and whether it is international relevance. Especially neuroimaging research are highly international relevance, and people are talking about these things. And this is a very, very active type of platform. And if you can do this type of research, definitely you can climb up in the ladder of academic, right? And then duration of study is very important. Some people do research and okay, this has to be finished within 3 years, 4 years. That's actually, it is not acceptable. In my way, actually, what I'm doing is if I can can find good results in my research, then if it is need to be, I mean, extended, then I have to ask for extensions. It doesn't matter. I mean, it should not go with the time. I mean, we should not strictly adhere with the time duration, right? And then targeting low impact journals is very common in Sri Lanka. I mean, I have seen even in professors, they are publishing papers in very low impact journals. So that's not good actually. As radiographers, we can reach high levels if we know this knowledge. I mean, if we can gather this knowledge, especially in the field of neuroimaging, right? And then poor and lazy writing is one aspect, and people are not happy to write more, and then actually that is very common thing, can be experienced among our colleagues and or other within the category of radiographers, and do not acknowledge the contribution is one other aspect, right? And once you get support from some funding institution or any other place or any supportive collaborator, then we have to acknowledge them in the paper, right? And then plagiarism, we are detecting so much plagiarisms when they submit abstracts. The best example is this recent one. I mentioned that we got even qualified. I mean, even the academia also, they are taking some. We noticed some plagiarism or similarities in their abstracts, right? That is totally unacceptable, right? The last point is scientific misconduct. Means that I have seen some people, they are showing, they are in their profiles, you know, that as an example in Google Scholar profile, they are showing the research, but they are not belongs to them, right? They have taken it very carefully, they have taken very smoothly, they have taken from some other person, and then they are showing them as their task. So that's totally unacceptable. That is called a scientific misconduct, academic misconduct. Those are not acceptable. Please don't do such kind of things as radiographers or any research, right? Okay. Okay. Now I'm coming to my topic today. It's actually I wanted to talk about more about actually the research techniques or otherwise research methods which we can incorporate in MRI, because I'm sure the people who are joining with this webinars, they have experience in MRI. They will definitely understand what I'm telling, and they are working with the mouse, means that they are handling data, but they don't know how to. Many of them are not much aware about how to use this data. That's the major problem. Then we should be able to use, and if we can use this data, definitely we can produce high quality research. That's what we want to introduce, right? I have taken one example here. If you do a research, definitely you have to concern two aspects of research, right? One is the novelty, the other one is the usefulness. I have taken one example. This is my example, actually. You can produce the same pencil here. I have given a pencil here. You can produce the same pencil in different shapes, right? Different shapes, right? And but actually, there's if I ask a question from your audience, that sometimes you will say, okay, there is a novelty. It doesn't matter. You have taken into account the novelty in this production, right? But can we see any usefulness here? You cannot. We can make in different shape the pencils, but you can have actually novelty. But the problem is whether it is, we have a problem whether it is useful or not. If it is not useful, no point of producing something novel, right? So that is the case, that if you want to produce something, please ensure that you have your production, or otherwise your research findings should have a novelty, and also at the same time you have to check whether there is a usefulness or not, right? This is the major concern when you do research. As good researchers, you should always follow these two concepts. Okay, right? And these are some of some application, actually neuroimaging research. I'm talking about always when I explain this slide, you will understand. Okay, are you going to talk about the brain? Yeah, I'm going to talk about the brain, because neuro means always the central nervous system, but it is not only belongs to the central nervous system. It is also has peripheral nervous system. But here in today's lecture, I'm interested in the central nervous system, especially the brain. Okay, we can use neuroimaging research. We can use to diagnose or provide treatments, or otherwise to detect conditions, detect the lesions, or otherwise detect abnormalities in these kind of conditions. But this is not limited to this kind of conditions, but there may be so many conditions rather than this mentioned condition, because I have some experience in this condition. That's why I'm taking this example. So one is schizophrenia. You can see some people having schizophrenia. You can have with psychosis, that the schizophrenia is more scientific word actually. In the day-to-day, and sometimes you might heard the word psychosis, psychotic people are available. There is a problem of in actually neuroimaging research can be used in psychiatric diseases. That's why I have taken two examples here. One is schizophrenia, the other one is depression. There may be some other conditions like the bipolar disorders, and then anxiety, and then some other there are different types of depression, and then first episode is schizophrenia. And likewise, there are so many a variety of conditions in the field, right? In the health setting. So why do we need to conduct research? Is we can find information or abnormalities of people having schizophrenia or depression or any kind of condition, right? Using neuroimaging research, and then using neuroimaging research, we can find abnormalities of Alzheimer's, and then we can find stroke people. What kind of arrangement of this their brain is been having, like structural or functional or diffusion problems there in their brain, because stroke is a leading problem causes for death in at present. So, and in addition, we can apply this technique in diabetes like condition. Diabetes is more associated with digestive system, but it is also indirectly associated with the brain. So people have found people having diabetes have some structural changes in the brain, and also functional and diffusion or perfusion changes. So likewise, we can apply neuroimaging research in the based on MRI in a wide variety of conditions, right? And then sexual deformation I have taken in the right lower corner, you can see, means that you can even forensic medicine, I have seen people are using some manual, people are taking some manual measurements to classify people, gender-wise classifications, right? For gender-wise classification. But using MRI, we can go some further investigations using neuroimaging techniques, right? And then to classify male and female people. And then normal aging. That's also very interesting among neuroimaging community, right? Because still people are searching for how brain is aging, means that starting from gestational stage, and it is end up with the death, or otherwise adult, and we can find how this brain is evoluted throughout the period of lifetime. So likewise, we can find normal aging, right? And then we can also apply in pain kind of condition. I'm taking few examples, you can apply in many cases, but I have some experience in these conditions, right? Especially in the pain, means that that someone can complain, I have a pain. Someone can some other one can complain that you don't have a pain. So how can we prove this? This is one example. I came from China actually, right? So my professor had to find a solution for this, because the one court of China has requested to find a method to find out how they don't know that court has asked a question from culprit. That's they haven't they don't know what actually these people are telling, truth or false. That's a problem. So they have introduced the neuroimaging to find out whether these people are telling truth or not. That is actually beginning of China, but not applicable to other countries. The pain is also can be applicable, because there are pain cortex in the brain which is govern the pain, which can govern the pain of the body, and also can be useful these regions to for treatment as well, right? And then tumor imaging also, that MRI based tumor image is very popular. You may have seen, and you can find how these tumors are associated with the other regions, especially functionally and structurally, right? And then comes to the most familiar one. These days we are working on epilepsy studies. Epilepsy, that the epilepsy people are observed with diseases, and sometimes there are some the wide variety of epilepsies are available in the health setting. So you need to find the abnormalities of epilepsy compared to the healthy controls likewise. And in all these cases, we can use these findings or abnormalities to produce biomarkers to predict people or protect people from any possible health conditions, right? So in that case, we have to use some new techniques. We call them as neuroimaging biomarker development, right? In this case, we have to use machine learning techniques. Machine learning techniques means the machine learning these machines can classify people based on information found from this wide variety of techniques, right? And usually for neuroimaging biomarker production, we have to find the structural abnormalities we can use, and then we can use the functional abnormalities and the diffusion abnormalities and then perfusion abnormalities. Likewise, we can find the abnormalities first, and then once we find this abnormality, we can feed this information to a computer. This computer is called training computer. I'm using this word because you can be very, then you can nicely understand this. Okay, and actually those algorithms, I'm taking the word computer to make you aware of and make it is very simply for you. Okay, we are taking information from patients and healthy subjects, and then we are training computer, and then we are training the computer, and then if you have new volume of information or otherwise new information of some unknown person, then definitely we can use this information to test using the testing computer. Means that this computer is rich with information of patients and healthy subjects. If you know new information, if you know if you have information of, but we are not sure whether this brain belongs to patients or healthy controls, then definitely we can go for machine learning, and we can go for more advanced techniques to classify these people, these conditions, right? So in that case, we have to use structural, functional, diffusion, and perfusion imaging. But these are not limited to these cases. But I'm talking about the basic techniques, right? Okay. Yeah, I'm jumping into the very first the technique, which is we call voxel-based morphometry method. Yeah, I'm taking time. Voxel-based morphometry method, which can voxel-wise compute, which can compute voxel-wise the information of human brain. I have given one image here. You can see left side one. This is a T1 weighted image. By looking at this, you can see that if you take one particular place, you can have three different kind of tissues here. One is gray matter, white matter, and CSF. That can be either tissue type. Okay, always. So if you are interested in counting or otherwise quantifying the information of this concentration or volumes or thicknesses or such kind of information, you can definitely go for voxel-based morphometry. This the Professor Carl Friston, actually we consider him as the father of neuroimaging. He has today he on 278,000 Google Scholar citation, because he's a theory maker. All the people, all the researchers in the world using this person's the findings. He's a theory maker, that's why he's getting this much of the citations. So in this case, in the voxel-based morphometry, what we are doing is, I'm not going to explain everything, but I'm trying my best to make you aware of this technique. The 3D T1 image is required, and then what happens, we use this image for further analysis. Means that the T1 image can be used to divide into several three components, major components. I told you, if you look at one particular voxel, it has a component of the gray matter and white matter and CSF. So therefore, we can nicely segment this into three different categories, three different compartments, right? And then before that, it has to undergo some pre-processing steps. We call them as a noise correction, bias correction, and then intensity normalization. And then you cannot use with this image with scarp. You have to remove the scarp. Likewise, we have to take separate volumic or concentration or information, and then using this information, if you look at one particular place of this image, there is a value. This value means if it is volume, it is the volumic value, right? And then you can use these volumic values for group level analysis. Group level analysis means that we can use statistical testing to do the group comparisons, to do the group comparison. There we can apply t-test, ANOVA regression, and etc. Means that we can apply any kind of statistical techniques, right? In this, you have to remember, what you have to remember is you have to partition the tissue types as gray matter, white matter, and CSM. Then you can compute each the values for each voxel, right? Okay. And then when it comes to tensor-based morphometry, the major difference is here we are interested in the whole volume of tissue, not segmented tissues as same as in the previous case. Here we are interested in gross volume changes. Gross volume changes means we do not segment the tissue types into different partitions, right? But actually, this is a very nice technique. We are going to publish a paper very soon. I mean, this one actually I'm sure you can understand very clearly. What we usually do here also, we should have T1 weighted image, and then we use this image to produce, to find out the gross volume changes of human brain. Here you can see actually the template. We have to use a template here, and then you have individual brains. If you are recruiting people for your study, you have the individual brains here, and then you have the template. This is a common template. What we usually do is we use this template to transform this template to individual brains. We are transforming this individual brain into the template into individual brains, right? And then there we can find how these individual voxels are deforming one by one. So therefore, finally, we can get deformation fields. That's why it's called the tensor-based morphometry is a variation of deformation-based morphometry. Means that we are depending on the deformation fields. When you transform, just understand this, very simple. The template is transformed into individual brains. And when you transform, you can see that some brain voxels are deforming into that individual brains. Means that we can find what is the amount of the variation happens there. Okay. Likewise, or otherwise, you can, if you are interested, you can do the other way around. Means that you can transform this individual brain into template. Usually, what I'm doing is I'm using both techniques, right? And then, right. Someone is disturbing me. Okay. Right. And then after that, this gross volume changes can be used to take decisions with statistical analysis. It means that once you have this information, remember that you don't have only one information. If you take one volume of voxel, you have information of three-dimensional information, so X, Y, and Z directional information. Therefore, we call it is multivariate information. For multivariate statistics, we cannot use just two sample t-test. Therefore, we have to go for multivariate t-test. Multivariate t-test means that we call them as Hotelling t-square test. That's why finally we can get this t-square map, right? Finally, we can use this t-square map to produce, to compare brains for the gross volume changes. Right. The third one, under structural imaging, you can see that I have given one here again, or it is also T1 weighted image, but it is sagittal view. You can see that if you are interested in cortex of brain, you can reconstruct the cortex using this T1 weighted image, right? This reconstruction process involves more than 30 steps. It's a huge step. Usually, if you use the free surfer like software, it takes for one subject, it's usually one day. I mean, we have to spend a lot of time. Depending on the technique, actually, free surfer uses a technique like deformation kind of technique, means that it is a little bit advanced. That's why it takes more time, right? If you use CAT12 like technique, it compute the cortical thickness one by one, means that voxel to voxel, right? Likewise, there are different kind of techniques. Finally, we can find cortical thickness, cortical volumes, cortical surface areas, and cortical gyrifications, and sulcus depth. Like there are many metrics. I mean, structural metrics can be used to compare the human, the healthy and patient's brain, right? This is really interesting. If you are interested in structural imaging, definitely you can go for this field of imaging, right? For your higher studies. And I have given one example. This is graphical user interface I used in CAT12, means that computer and toolbox. I'm going to introduce toolkits after each and every technique. SPM is very common, and CAT12 is but a little popular type of technique because it doesn't require much computational skills, right? But when it comes to free surfer, you need some computational or programming skills. Therefore, if you are not interested in programming, you can go directly to computation anatomy toolbox, or otherwise CAT toolbox, right? So these are the techniques. Later you can go for searching of this toolkits. And some related work I have mentioned here. The first one we published 2018. This is actually the abstract, and then again 2020, all three the abstracts were published in OHBM, which is the major platform. If you can publish your abstract in OHBM, means Organization for Human Brain Mapping, that is great. Actually, that is actually really great. I mean, as radiographers, if you can publish your abstract, you can get more comments from them, and that is the major platform in the world who gather all neuroimaging researchers gathered at the same time for actually in every year, right? We should, if you can forward abstract, if it is accepted, definitely you can go and attend. You can hang around with them. That's really important if you are interested. Okay, and the most recent one, classification of a patient with epilepsy and healthy subject using structural MRI tensor-based morphometry. This one I explained will be published soon in our IRC one. Okay, and then I have reached impact factor 4.3. You can this is the best evidence for you. You can, as radiographers, you can reach up to the level of 4.3. I have reached that level, means that there are no limits. If you are highly committed to research, then definitely you can reach that level. It doesn't matter, right? Okay, the first chapter is closed. I have to talk 4 chapters here, especially the functional MRI started. You might have heard this one definitely, but I'm sure you don't have much experience, because this is more associated with the surgeons. When they are interested in one particular place of brain, they usually conduct in hospitals, but I'm talking about research aspect. From the aspect of research, Bolivia is the first scientist found this functional MRI technique that is actually based on the gadolinium bolus injection. That is not the way that usually, but in current practice we use blood oxygenation level dependent MRI, because at the beginning they didn't have this technique found, and the Ogawa in 1992 found the blood oxygenation level dependent MRI, means functional MRI doesn't use any contrast media anymore. Okay, and you can see the first paper here in 1992. If you look at this image, this Ogawa has found, when you perform a special task, okay, as an example, if you can give a photic stimulation or otherwise visual stimulation, then definitely when you look at that stimulation, or when you see that your occipital cortex is activated, right? You can see how he found in the bold imaging, blood oxygenation of in this area is increases. It utilizes more oxygen. That's why the signal is very high. And then when here in the C image, you can see that the color coded image gives in some areas gives more intensive the signals, means that those regions might be highly associated with visual stimulation. So this is how the functional MRI coming into existence. And in 1991, actually they have used gadolinium bolus, but when it comes to 1992, this is a greater, actually very nice breakthrough of findings in neuroimaging. The people have started working on functional MRI based on ball imaging after that, right? And then comes to I have some example here. If you do finger tapping, I'm talking about functional MRI. If you do finger tapping, definitely the responsible part of your brain is activated. You can see here when you finger tap, when you tap your left side, left hand, and then you can see the opposite side is activated. If you do the other side, the opposite one is likewise you can see that opposite part is activated. We can catch it, right? If you have a condition or if you have any disease in the brain, then this you might have will not experience this information because of the condition. The second image gives such kind of problem, right? Such kind of scenario, right? If you can, then the people have found how can we measure this neural activity. If we should have a method to find this neural activity. So they have found blood oxygen. I told you, when you perform a task, definitely you can see some blood oxygenation consumption is happening there, and then blood flow is increases and blood volume also increases in that place, that particular place, and changes in metabolism also occurs. So using this technique, they have started to find new techniques to find out this functional MRI advancement, right? And then I have given here one example: resting and activated. When you do some stimulation, as an example, if you are looking at visual stimulation, or otherwise we can deliver some pain stimulation, or likewise when you do something like that, that particular area neural activity is increases of that particular responsible area of the brain, right? And then what happens, the vasodilation is increases, means that cerebral blood flow is increases, metabolic rate of oxygen is also increases, cerebral blood flow, blood volume also increases. And finally, this is huge mechanism. Actually, it's very difficult to explain within this limited time of period. You just remember when something happened like that, when there is any specific place in your brain responsible for some specific task, remember that there is a change of ratio in between oxygenated blood and deoxygenated blood. Because of this reason, the deoxyhemoglobin level goes down in that particular areas, and then finally what happens, the MR signal is increases in that particular area. That's how you see in that particular area you get high amount of signal compared with the other regions, right? But remember, in functional MRI we do not use T2 or T1 information, but instead we use T2 star information. Those are a little bit, actually the signal wise it is a little bit low, the resolution is a little bit poor, right? We will discuss these things in next slide. The usage of functional MRI is very important in clinical psychiatry. We use it to identify the people. You know that if anyone comes to psychiatric department with the condition, sometimes the clinical psychiatrist or otherwise the consultant might have some problems with this person having problem with the schizophrenia or anxiety or depression, or they usually complain they're in a problem of categorizing these people. That's why we usually produce biomarkers using images to classify these people. That is our part. If we can produce this kind of biomarkers, that will be greatly helpful for taking decisions by the health community, right? Okay, and then cognitive neuroscience is also applying this the functional MRI, how people perceive information and how do they remember and how they speak, what happens when they speak to the brain, and then what happens in the brain when they are thinking and then make decisions, and so when they solving problems likewise. So we can find this information using the functional MRI, right? And the pre-operative planning is very common. Also, actually, you are very well aware of this. Before they perform, I mean, surgeons before they perform the surgeries, usually they are very happy to see what kind of region is this. If this region is removed, or removed from this person, then what happens? What will be the consequence? It means that we should have an idea about what is the function of this particular area. So that's why we need this functional MRI. So this is really important, right? And then structural MRI versus functional MRI, that there are major differences. One is single images taken in structural imaging, but in functional imaging, we are interested in a series of images. It means it takes time, right? When you do this kind of research, the patient has to stay longer time of period within the scanner. So that's one bad side of this type of research. But we have to give proper instructions to keep them immobile, and we can definitely collect a required number of volumes, right? Usually in our practice, in this kind of research, we have to produce, we have to more than 400 volumes in a particular subject, right? If you are interested to perform functional MRI based research. The first one is high resolution. That's true. But when it comes to the functional MRI, it is low resolution. It doesn't matter, but it is signal rich information. But when you look at, you cannot take decisions by looking at these images because it is low resolution, nice image, right? And the task fMRI image, if you look at this, you can understand when you do perform, this is a nice example. Actually, when you do perform, we have this experience in China. We have performed this kind of studies. The patient has to lie down in the scanner and then they are looking at the mirror. So you know that we can, the bird cage coil, it has the mirror, then mirror through mirror, you can get the reflected the stimulation from the video screen. It's really interesting. And then you can, you should have a nice design. And before you perform this kind of studies, you should have a nice design. This is called block design. Here you can see what happens when this person is open eyes and close eyes, right? When they open eyes, the stimulation comes to identified by the eyes, and then it goes to the brain, and the visual cortex is activated. You can see how bold change happens here when they open the eyes. Because of this spotting stimulation or otherwise visual stimulation, this brain area is highly activated, right? This is not limited to this block design. There are many kind of designs. If you are interested, you can apply these techniques. There are many papers available to learn these things, right? And then when it comes to resting fMRI, remember this is really really simple to understand. Although you are delivering some stimuli to the patient or subject in a task fMRI, remember in resting fMRI we do not deliver any kind of stimulation. What they have to do is they have to stay in the scanner by looking at this kind of white cross, or otherwise we can ask them to close their eyes, or otherwise we can ask them to close, just open their eyes, right? But we have to particularly give them instruction not to think anything particular. Means that if they are thinking, if they start to thinking about something, means that brain is activated. That's something hindering our findings. So therefore, we have to look for this kind of things very carefully. Usually, the people in the world, what they're doing is they usually ask the patient. That's very easy. They ask the patients to close their eyes. That's very easy. So even at this level, even without stimuli here, but remember the brain is activated. Based on this information, we can go for further analysis of how patients and healthy controls brain is activated. Like there are many more metrics to see these things, right? See or quantify these measures, right? So this is really important. Remember, we have major two components of functional MRI. One is in research, one is task fMRI, the other one is resting fMRI. Resting one is a little bit easier than the task fMRI. For task fMRI, we need much more the computational programming skills. And if you are interested in functional connectivity, just focus on this image. I'm going to give some, actually this is really important to understand. Many studies are based on functional connectivity. Once you collect this functional data, this can be either task or resting information, it doesn't matter, right? So what you have to do is, if you have a series of images, you can take two regions at a time, and then we can, here I have given two regions, one is blue, the other one is red. If you look at the blue region in each and every volumes, you can make a graph like this, okay? For each volume, we have a value. For each volume, we have a value. And likewise, we have nice time series. We call them as a time series. Then again, for red region, we have another time series. Then what we are going to do is, using these two time series, we can produce the functional connectivity, or we can find the dependency in between two regions using correlation coefficient. That's why it's mentioned in CC here. Using Pearson correlation or partial correlation, whatever the statistical method you can use to quantify the regional dependencies in the brain, right? We call them as a functional connectivity. If you can, the parcellate, first you have to divide the brain into different regions. That's why it is called brain parcellation. I have given an image. that the numbers might be varied. Some people have found 90 regions, some people have found 1024, means that that is based on these atlas are highly universally acceptable atlases. You can use this as atlases to quantify the functional connectivities. Okay. Then finally, based on these regions, based on this the dependencies of functional connectivity, we can produce functional connectivity matrix here. If you look at one axis here, you can see that this is if we divide the brain into 90 regions, you might have seen that there are 90 regions in this side, right? This direction, right? In y direction, again, you have 90 regions, and this color, what this color mention is that in each color mention the correlation coefficient or R value, right? R value, I'm sure you are well aware of these R values, right? Okay. And in addition to functional connectivity, we can find effective connectivity. Also functional connectivity, actually, they don't have any direction. They don't have a direction. We can just tell these regions are functionally connected, but we cannot say that this region is affected by the other region. Otherwise, the information traveling from visual cortex to temporal or temporal cortex to visual cortex. We cannot say like that. That's why the people have tried to find another technique based on functional MRI that is called effective connectivity, which has a directional relationship between two regions. This is one visual, this is temporal. Why I'm telling these things mean that when you compare normal healthy controls or the patients, definitely there are differences in this information traveling and functional connectivity. So therefore, we have to look for this we have to address this problem using these techniques. And finally, we can statistically compare this information, right? Okay. And then these are some advanced the metrics. I'm not going to explain everything, but remember, using these techniques, finally, you can find many number of metrics. One is actually the functional connectivity FC and effective connectivity REHO, LLFF. Likewise, in addition, we can take some network topological metrics. There are so many metrics developed by the people. Actually, these days we are working on these techniques based on epilepsy data set we are having, right? Okay, and we have few few actually the publications related to functional MRI. The first one is published in 2020 in the IRC conference, the abstract, and then OHB in 2020 we published actually it has this multimodal, means actually here this multimodal means functional and structural both. That's why it's called multimodal, right? And then the last one is going to publish very soon, which is about the functional MRI, but we have used mild depression and healthy control subjects in the publicly available data set, right? The toolkits, I have some toolkits, very interesting, and data, actually, this is the first one is DPassword, we call them as DPassword software, which will be used for the, we can use this software to for image pre-processing. Before you process images, you have to do some pre-processing steps. One is, that we have to remove skull, and then we have to do size timing correction and the normalization, and then the co-registration, and there are so many steps, filtering, detrending. There are so many steps has to undergo in each method, right? So for that purpose, we have to use the DPassword, and Gretna is doing some can compute network related information, and the rest is another software we can use for many quantitative metrics. And then FSL is another one. FSL is not only specialized for functional MRI, but also we can use for the diffusion and perfusion as well, right? And then functional connectivity toolbox. This is developed by MIT, and this also can do many sophisticated work, means that the functional, structural, and I don't know whether they can perform diffusion as well. The brain in Voyager is I have very least experience on, but all the others I have experience on all the other except brain Voyager. So if you're interested, you can go through these manuals and you can use you can incorporate those things for your higher studies, right? Okay, and then I close the second chapter. This is the first one is finished, structure. The second one is function. The third one is diffusion. I'm trying my best to explain within the allocated time, actually running very fast at the time, but I'm trying to make you aware of these techniques. First four techniques is really important, right? So diffusion, the concept of diffusion has come from Albert Einstein, first introduced the Brownian motion. And thereafter, they have found that the water is governed, or the self-diffusion of water molecules follows the Brownian motion. If you look at the water molecules, we can experience that Brownian motion because it's traveling everywhere, which can freely travel in the medium. That's the case. Okay. Based on that, they have developed diffusion MRI. You can see that. I don't know whether this is much clear or not. I just copied from internet. And the first image is given diffusion weighted image, and second is also diffusion weighted image. But you can see that in the second one, it is a little bit abnormal, means that there are some water restricted regions, means that water traveling is restricted, means that the signal intensity is very high in that case, means that if you use, usually we use gradient echo pulse sequence for this purpose. You can see here, just look at my cursor here. We are interested in mobile and fixed water molecules in diffusion MRI. You know that when you apply 90-degree gradient and then again apply 180, when you apply 180 gradient, you can see that some hydrogen protons traveling started, and then again in both cases it started, which I'm talking about the fixed water molecules and mobile water molecules. But remember, when you apply the 180 pulse, what happens? The traveling water molecules does not experience the second rephase gradient. That is the problem. Because of that, we cannot accept high amount of signal from moving water molecules. Therefore, you cannot see the high amount of signal in moving water molecules. Therefore, if there is any restriction of water molecule traveling, that's why it is appeared in white color here. You can see in diffusion weighted image. Okay, this is the very simple way of explaining this. There are so many things to discuss in this one, right? Okay, and then based on this information, they have started to find diffusion tensor imaging. All these techniques can be used in our research, right? So what they have found is that CSF, you know, it's like water. I mean, it has many water molecules which can freely move in the medium. And then in addition, we have gray matter and white matter. If you look at the white matter and gray matter, there are some restriction of movements. Okay, when you have some restriction, actually you cannot freely move these water molecules through the medium. Therefore, we call such kind of diffusion as anisotropic diffusion. When it comes to CSF, it is totally different, opposite, and then they can freely move in the medium. Therefore, we call them as isotropic diffusion. Based on this information, if you look at white matter here, I have focused specifically the white matter here. If you look at white matter bundles, remember that you cannot cross the walls or these neuronal walls, or otherwise the intrinsic, this intracellular space is there, and then you cannot cross these lines, or otherwise these walls, right? It can only travel through one specific direction. That's why it is called anisotropic diffusion. This can be experienced only in white matter, but not in gray matter or CSF, because that is the basis that they have used this technique, which is called diffusion tensor imaging, right? So we assume in diffusion tensor imaging, we are always talking about voxels. One particular voxel may have one directed kind of these are white matter bundles. Okay, we have more than one neuronal bundles, and means the neurons means that it's a bundle. So this bundle can be directed using this diffusion information. We can provide a direction for this particular voxel. And then the second voxel, you can see that there is another direction. For the third one, we have some other direction. Likewise, we can develop mathematical models. This one model, if you take one model, you can see here this is an ellipse, and this ellipse gives information of eigenvalues and eigenvector. Eigenvector is that the direction or the direction of movement of that particular voxel, and the eigenvalues means there must be a value for that one, right? Means that anisotropic value, right? Or otherwise diffusion value, tensor value, right? So we should have those two. One is the direction, the other one is value. So using these values, definitely we can quantify nicely the brain, how these tracks are arranged in the brain. You know that the first step is acquiring diffusion weighted data, estimation of fiber orientation, I talked already, and then application of tracking algorithms is very important, right? Using there are some algorithms, we can, don't worry about that. Those algorithms can catch these this direction. I told you for each voxel there are some directional, direction and also a value. So using this the tensor values, you can nicely track these, nicely catch this tracking. Okay, we call this as diffusion tensor imaging tractography. Means that finally you can find nice tracks in the brain of controls and also patient. You can see that sometimes you might experience in Alzheimer type patients there is a huge degradation or otherwise degeneration of white matter. You will be experiencing this kind of brain. This kind of brain is some white matters are missing here, means that those are degenerated. So this is really interesting. I'm sure you can work on these things, and this is all the sources are available and all the toolkits are available freely, and you can go for this one. And the very first image I will explain, this is diffusion weighted image. The second one is diffusion weighted image is not diffusion tensor actually, but it uses roughly 3 to 6, actually 6 direction. If you are interested in DTI, definitely you have to go beyond 6 direction. Mean that you have to apply more gradients to catch the more accurate directions. As an example, 64 like directions will help you to catch all the directions in the white matter tracking. Okay, and then the second one is ADC, actually apparent diffusion coefficient. Nowadays they are using the term as the mean diffusivity. This is completely opposite of the diffusion weighted imaging. Diffusion mean diffusivity means that CSF has more diffusion capability, therefore it appears as white here, but relative to other tissues, right? When it comes to fractional anisotropy, this is another metric we can find in diffusion tensor imaging, which explain about white matter integrity. White matter integrity means that you can see nicely these tracks are available here using color coded. FA means fractional anisotropic information are used here. Further, you can use this for statistical testing between groups, right? Okay, and then we can go for the functional connectivity. I have already explained here. As same as that, we can find structural connectivity also using diffusion tensor imaging. Here we involved the functional MRI. Here we involve diffusion weighted MRI. We can use this both information for our research. These are some toolkits. One is Panda, TrackWiz, and DSI Studio. These are available freely for you. And then I have taken one graphical user interface here, GUI. You can see Panda tracking option here. It is very simple. And if you go for further with Panda, you can see this kind of window. You can see that deterministic and probabilistic tractographic methods are available. You can nicely incorporate these things for your research. And the third one, 3 chapters closed, and the fourth one, this one, and this is perfusion MRI. If you want to measure the perfusion of human brain, means that blood perfusion. And the previous day, what they have used is contrast media, but nowadays we don't want to use any contrast media. We can use some labeled the blood, or we call it artery spin labeling, to label blood to produce this kind of images. Finally, you can get this type of the perfusion images. The technique is very simple. You have to have T1 image first. We call it a reference image. And in addition, we have to have the labeled image, which means that we are exciting some portion of, that blood moving upward here, and then we make a slab here, and then we excite some amount of the hydrogen protons, or otherwise the labeled, or we call it labeled hydrogen protons, and which can reach the brain parenchyma, or otherwise brain tissues. And then finally what happens, the first image is here, we don't touch it, and then again we have labeled image, means that when you change the labeling protons, by labeling the protons, what happens, the signal becomes reduced. Okay, now labeled image is a little bit reduced in signal, and then in addition we have reference image. So you have to make some computations here to reduce the labeled image from reference image. This is what we usually do. Remember, this is not the minus sign. Actually, you have to reduce the label image from reference image, and then finally you can find how the perfusion information can be seen in our brain, right? And these are the 4 major techniques. I'm not going to explain all these things now. And those 4 techniques finished, and then I will quickly go through this one, quantitative MRI. These are the MR relaxometry you can use. You can use these techniques, the T1 weighted image, T1 weighted information, T2, and the PD information, and T2 star relaxometry. You know that for a particular voxel, we can find T1, T1 relaxation, and then T2 relaxation, and T2 star relaxation, and PD. So we can use these images. You can see this color code here. This color mention the value of each T1 time, right? So using these maps, we can definitely quantify, right? Quantify, we can make some decisions, right, on the group level statistics. And what is important here is that if you are interested in PD here, if there is an increase of PD, means that free tissue water are there. If there is increase of T1 values here, which means that there are the water content in that place is very high. Likewise, I have given a nice characteristic of each metric. You can go through this leisurely, and please apply these things in your research. And there are some toolkits for even for this one, FLM maths, FSL maths, and then HMRI tools also can be available, which is running under SPM. And you can use some data to process this kind of technique, right? Images. And the MRI spectroscopy, I'm quickly going through because I'm running out of time, I think. Because I have explained very clearly major 4 techniques, but there are some other techniques also which will be incorporated in future for your research. So that's why I'm explaining. The MRI spectroscopy is to analyze chemical composition, means that you know that usually we produce images using hydrogen protons, but in MRI spectroscopy, we use some other technique, means that we can resonate, otherwise we can excite not only hydrogen protons, but also we can excite some other protons as well, like sodium, potassium, like protons as well. If we know the Lambda frequency, definitely we can get them excited. Means that we can find their information, and then we can make spectrums or graphs, and then we can compare using groups of statistics. And especially for this kind of one is applied in tumors. And you know that when tumor is originated in your brain, what happens, the chemical composition is extremely changed. And therefore, we can see how the information of this particular region is vary because of this the technique. Using this technique, we can definitely quantify this information. Remember, very simply, you can remember that this technique, we do not only excite hydrogen protons, we can excite some other protons as well. And using these techniques, you can finally identify the peaks. Here you can see some peaks here related to sometimes amino acids, sometimes choline, sometimes naphthalene acetic acid. Likewise, we can find some different information, right? Concentrations of different information, right? So this can be applicable in many cases. I have mentioned a list of cases, and actually tumor, stroke, epilepsy, and some other cases, right? Okay. And then finally, actually there are some, this is also very important. If anyone is interested in doing meta-analysis, meta-analysis is very common, something very common in our society. Many people apply, not only the MRI-based researchers. You know that the images, there are some databases. They have the information of already found research. So what we can do is, if you want to take collective decisions on this, if you're interested in languages, okay, let's say you want to find the area of responsible for language, okay, you have to find this one. Then you should have to find all the information, all the literatures available in the database. So it's very difficult to find one by one. So therefore, we have to go for this kind of databases. Someone has searched here. I can give you one example for languages. 1,101 studies available. Using this available data, definitely you can go for collective decisions, mean that you can take decisions on how the brain is activated for languages or responsible for languages. Likewise, we have 2 databases. One is Neurosynth, the other one is BrainMap. If you're interested, you can go and see what they are doing using this available data. We can take some conclusions on some studies have some disparities, otherwise inconsistencies. So therefore, in that case, we have to use this kind of techniques to take some collective conclusions, right? And some other types, I will quickly go through these techniques. EEG also can be incorporated in MRI or neuroimaging research or brain research. One is electroencephalography, means that you can using these electrodes, it is a cap of electrons. You have to wear the cap, or otherwise the subject has to wear the cap. And this cap has some electrodes which can be connected to a scalp through some conductive the liquid or something. And then we can definitely detect what happens when you, even at your resting state, there are some electrical signals are generated. Using these signals, we can find the information of patients and healthy controls. Okay, and then comes to positron emission tomography. Also can find some info because it gives some information of the metabolic information, right? Metabolic information, how some areas are metabolically active. Because in PET, we usually give the glucose. Depending on their consumption, we can produce images, and we can incorporate the PET images and also fMRI or EEG, whatever. We can take this all together and can make some collective decisions, right? And then MEG, the last one that I want to explain. This is magnetoencephalography, means there are some very sensitive the machines. This is highly expensive. This expensive type of machine can catch, our brain is always active, some electrical signals are producing, and based on these electrical signals, the magnetic properties can be changed. So what this machine does is that they can catch these magnetic properties based on these electrical signal changes in the human brain. So based on that, also we can find information. So finally, actually, non-human primates imaging also can be applicable. Actually, I'm really interested in this kind of research, but we need to have much more funding for this type of research. No one has conducted so far in Sri Lanka. So primate data exchange, which is the one recently established platform, we can find data from them, and then we can analyze, which is freely available. And then National Chimpanzee Brain Resource is also available, and Japan Monkey Center. And then if you're interested in imaging chimpanzees or monkeys or rabbits or rats, definitely you can go for that. Means that with funding, and I mean that it's very simple. What you have to do is you have to have some RF coils like this. You have different size of coils, right? Here you can see where there are very small type of coils also. You have to put or place your rat or rabbit here with anesthesis, and then definitely you can image. Then you can find information as same as in human brain in the MRI scanner, right? Okay, you can find a nice evolution here. It is given here from marmoset, and then starting from that, it is end up with human. You can see how this the structural information are gradually evolution is happening, and then you can see that this structural information, we already talked about the structural mechanism. How do we find it using diffusion tensor imaging, right? That is also can be incorporated. Only thing, we have to keep them for a longer period of time. If you are applying different types of technique, if you are usually when in practice, actually it's not that easy. Means that first we have to perform T1 weighted image, structural, and then functional, then diffusion and perfusion. It takes much more time. So we have to have, even though they are patients, we have to keep them well incorporated, otherwise very difficult. Okay, and then Martin van der Hol is the very famous guy who is doing research in chimpanzees and human brains. So he's comparing how these mechanisms are arranged. You can see this completely, I mean, the close to human brain, their mechanism. And finally, he has found in very recent article, people who are behaving like, psychotic people have some same kind of arrangement with chimpanzees. Chimpanzees brain arrangement and functions quite similar with psychotic people. Means there are something to consider. I mean, very interesting finding. You can search on the internet for more details. Okay, this is one actually the machine learning part. Actually, if you are interested, only you can go through this slide.
the MV multivariate pattern analysis is very important for this developing biomarkers for our studies. Once you have to find abnormalities, and then you have to go for the machine learning to classify or do predictions, right? This is a nice example. I'm always taking this one. This baby is laughing here, and it's, in this case, baby is crying. At this, in these two conditions, brain function might be vary because of the pattern of function might be vary. So in that case, this type of patterns can be evaluated using multivariate pattern analysis. What we have to do is, here I have taken only two voxel, voxel 1 and voxel 2. And from this one, I have taken only two voxel, but with values. It has values, functional values or signal. And then for the second phase of condition, I have taken two other voxels as well. Using this information, we can nicely plot this graph. As an example, 1, 5 means the first axis, voxel 1 has one axis, voxel 2 has the second axis, means that if you have three, if you are going to consider three voxels, you have to have three-dimensional arrangement for this one, okay? Likewise, for 1, 5 means here 1, here it is 5, you can make a plot here. Likewise, we can nicely plot this information. If we have more data, this is actually only two conditions, but you can have many different babies, and you can consider two conditions, and you can plot nicely here. And here actually you have so many babies, okay? And then you can make a nice boundaries here, and then you can develop a model. Okay, using this model, you can nicely predict people, they are laughing or crying. Likewise, we can make predictions. Okay, this is a simple example. You can access more in the internet. Okay, right. For machine learning, I have given some assistance for you. You can access these toolkits, especially pattern recognition for neuroimaging toolbox, very popular, developed by, actually it is developed by London University, and then University College of London, and then we call it as Pronto, very famous term, which is Pronto. And then MBPAN is developed by our lab in China. We developed this, very useful. This is very user-friendly software. You can use your information for machine learning type classification or clustering or predictions or regression kind of analysis. Likewise, we can use any kind of techniques with this software toolkit. Okay, this is just the last part of my lecture, and the citation, this is about just because, you know, my students, there are so many students and colleagues, they might be wondering what is, always we are talking about citations. Citations means once you actually, it is a reference to source of information used in your research. Once you publish a research, if you do not get citations, means that that is not useful for the society. That is not useful for the society means you can see this person is on more than 280,000 citation, means these are his publication. I have taken only 3 publications here. If you look at this, the first one, statistical parametric mapping, this is a major breakthrough found in 1994, and this publication has got more than 10,000 citations, means it has been referenced in many articles more than 10,000 times. This is what it explains, right? So if we have five citations for one particular paper, means that it has been cited in 5 places in the internet, means that it can be countable, right? So you can see how in each year he has earned his citations here. Okay, and then total number here, you can, we call this as the Google Scholar citations. The academics are really interested in earning citations because they are getting marks for their promotion, and this is really interesting to know. And the people who do not understand, people are not aware of this one, can nicely understand what I explained here. Major platforms for I have not much the techniques onwards. Now onwards means that I have a few slides to give some information I can quickly go through. And major platform for imaging data, the COINS, which has the schizophrenia data, and ADNI, definitely you can get some Alzheimer-based the data, and connectivity. This is a huge database, a human connectome project related database you can find. And the Open Neuro, always I'm using the data set of OpenNeuro, which is very comprehensive type of data set. I mean, not only the images, we can find the image related information, clinical information, and the patient is more information as well. Okay, and SchizoConnect can also find information. IXI data set, which is a little bit older, but you can still find some normal healthy data set. Okay, and RFMRI network, which is hosted by the China, and also you can find information, find data from that website. Okay, and major publication platforms, Organization for Human Brain Mapping. I told you, please try to publish your work in the Organization for Human Brain Mapping. That is the best place to publish neuroimaging research or abstract, because that is the only place gathering once a year all the scientists in the same forum. All imaging scientists are waiting for that forum, right? And ISM MRM is also something like equal OHBM, but here it is a little bit mathematical side oriented, or otherwise engineers are highly involved in ISMRM. And European Society of Radiology, I am a member of this, and you can get more. There are freely available webinars, or otherwise the video clips, and you can access. And ASNR also I am a member, and then I have gone through many videos. And then ISRRT, our mother society, and also we have there is annual meeting, you can publish your work there. And then Sri Lanka College of Radiologists, the annual platform, we can publish our research. And also in future, we are planning to develop SRTSL, and also to make a nice research platform using our scholars, and to publish our research in that platform. SRTSL is the Radiographer Society of Sri Lanka, right? And teamwork, I have done many teamwork in last 5 years. You can see my face in the back row. My professor is in the very, front row. You can see he's not that. He's very young man, and he's very knowledgeable and very honest and very generous person. And he's a professor in having more than 7,000 citations so far, just 4 years elder than me. And the Tianjin Medical University of China, which is my last experience. And then KDU, we are going to develop a research culture, or inculcate research culture in our community, or radiography lab, or our department. We are having some computers, and in future we are getting some computers also. And then you can apply for your postgraduate work. Okay, ongoing project. These are something about ongoing project. The epilepsy work is already going on, and then 2020 to 2021, we are actually I'm continuing my work, halfly done work with Tianjin Medical University, and which is about schizophrenia. And in 2022, I'm going to introduce new project. Of course, you can join with this one. I'm preparing proposal. This one. This is I will definitely incorporate these techniques. One is structural, functional diffusion and perfusion MRI machine learning techniques. Okay, I will incorporate these things. And finally, I have some messages for you. Suggestions to enhance professional recognition in Sri Lanka. Need more opportunities for higher education, especially, you know that we don't have master program. Especially, we need to look for master program. You know that like the science faculty in Peradeniya has started processing master program for physiotherapy, and likewise we need to have someone from Peradeniya or KDU, and take part in societies related to radiology is very important. We have to take part in all the societies, and disseminating knowledge among peers means like me. And once you obtain your PhD, no point of keeping in under your pillow. I mean, you should deliver your knowledge. You should share your knowledge with your colleagues or peers and or students, and we should inculcate this into a new generation. We should deliver this to do good. Expecting good outcomes. Okay, and then need more writings. That is the most hindering problem. People are very reluctant to do this writing, and then more institutions to produce quality radiographers. This is the one aspect actually we have to look for, because in Sri Lanka we have only two institutions which produces the graduate radiographers, but we have some other place which is the very first place which produces diploma radiographers. We have to develop it to up to the level of graduate radiographers, right? And then participation in conferences are very, very important. And each and every conferences we should try to attend, and we should try to participate, right? And then encourage lateral program to get them involved, already practicing radiographers, to do more research and more research publications we need. And motivation through incentives means that people have no interest to do research if they do not get anything after the research findings or the publication. So we have to improve them. We have to motivate them to actually, this message should go to our administration in hospitals especially, and we can introduce some intensive incentives or allowances for researchers to do more, get them involved in research. And then administration opportunities we should give. And then the terminate diploma program gradually means that we don't want any diploma radiographers anymore, because we are going far beyond that. We have many graduate radiographers. We have to improve them. We have to develop the profession. We should have, if we want to develop, we should have the graduate radiographers more. Okay, and then CPD programs means that continuous professional development programs like this should be involved, and they produce knowledge-seeking radiographers. We should produce knowledge-seeking radiographers rather than just following knowledge. That's how we can develop our profession. And then new circulars for research facilitations and MOUs with developed countries. Usually we should target developed countries because they are far beyond us, because they are still we are doing some very primitive type of research. When you just have a look, the other countries, they have moved forward a lot. Doing many high level type of research. Future of radiography is not that bad. We can expect much more. We can keep some expectations. We can keep some hopes. In future, we have 3 PhDs already, Bimali, Roshan, and me. And actually, in addition to that, we have upcoming PhDs, Pradeepa, Amal, and the Sachira is also from Japan. And one KDU student, Duminda, and Dilan is also coming from China in near future. And so therefore, I'm sure these people will guide the youngsters, and we will get together and we'll do something in future for the betterment of our profession. And I have one MPhil opportunity for one student under my supervision. This is funded by Kotlawa Defense University. One student will be selected from actually based on this qualification, minimum requirements, second class upper division above, and computer literacy and sound skill in scientific writing means that it can be increased gradually. We can enhance it when we do some work in our environment. So therefore, if anyone is interested, you have to, my suggestion is to go for more education opportunities. Please seek opportunities. Please go and do some work. Especially try to become qualified researchers and radiographers. If you can become researchers as becoming radiographer, that will be much greater, and you can deliver much knowledge for your youngsters as well. Okay, and one take-home message. This is actually, it's time to remove obstacles. It's time to remove obstacles. Actually, I am always trying to remove obstacles, because that's why I wanted in this actually this is a long-awaited lecture to deliver my youngsters, and I collected this information for a longer period of time, and then finally Bimali, Dr. Bimali invited me to carry out this process. I barely took this opportunity because I was extremely busy. Anyway, I'm so satisfied with this one, and it's time to do some research. And please show your colors. Please don't hide your capabilities. Please show your colors as radiographers. What I want to show is we can go beyond that the society usually expected from us. Okay, we should try to do something new always. Please remember two facts I explained: the novelty and usefulness. If you do research, please apply those two. Then finally, your research will be accepted by top class platforms. Thank you very much for listening to my lecture. And it's about actually 1.5, it's more than 1.5 hours. Actually, if you have questions, you can ask now. If you have any further, if you need any correspondence from me, and if you need more information, you can contact me via this the email. Yeah. Okay. Thank you very much for listening. It's about 92 participants remaining.
Thank you, Dr. Vasana, for your valuable and informative presentation. So now it is time to ask questions. You can use the chat box, or you can unmute the microphone and ask questions.
Hello. Hello.
Hello. Yeah, I can hear.
Yes, can you hear me? Okay. So it's really a great presentation. Thank you so much. I'm Silas from Australia.
Okay.
One of the main difficulties that the data collection in Sri Lanka, because if you go for neuroimaging research, you need to have data. It's almost confirmed data. Is the disease should be confirmed by the consultant psychiatrist or radiologist. Because I used this VVM techniques for my master project. Finally, I ended up with the research data, which is collected from the UCL, University College of London.
Yeah.
I went to the National Hospital and I checked the data, and they were doing just 2 cases of dementia per month. Okay. So if somebody wants to do this kind of neuroimaging data for the undergraduates, by using the data which is collected from the Sri Lanka is so difficult. So what is your opinion on that?
Yeah, actually, still, actually, we don't have that. We are always time, because I'm the first person, I think, we are working on this platform. I mean, I have to lead this kind of things first, because first I have started from our hospital, which is the KDU Kotalawa Defense University Teaching Hospital. It is our university hospital. We are gradually trying to collect our data, because you know that it's very difficult to collect data from it's very difficult to use our others' data. Sometimes we are missing some details, and if we have a chance to collect our data, why we are not going to such kind of collections? And you told that you collected already, right?
No, I finally used the data which is collected from UCL, University College of London, but okay, I failed to collect the data from National Hospital.
Means that you had problems collecting data, means you didn't get the permission, or sometimes
no, it's no data. It's if you want to collect the neuroimaging research in Sri Lanka, you have to wait more than 5 years to collect the data, reach the amount of data.
Okay.
Yes.
Oh, okay. Means that it is not readily available. I mean, you are getting very few number of subjects, right, at a time.
Yeah.
Yeah, that's the problem. Yeah.
Second one, I need to ask that. Say the methods you introduced, the BBM or tensor-based morphometry or surface morphometry. And I went to your research, and you told that the combination of the method is good, and it's around 85% of the prediction. So the 85%, is it clinically relevant or not?
Which one?
You in your research article, you published in the Neuroimaging Bulletin,
okay,
and you mentioned that the combination of this method, BBM or tensor-based morphometry or surface morphometry.
Yeah, okay. Just give me 1 minute. I'm going back to my one. I think you are talking about this one. Yeah, this one I think you are talking, right?
Yes, this one. Yeah. So you mentioned that it's finding that the combination of this method is prediction is 85% is
Yeah,
good.
yeah.
Is it clinically relevant or not?
Yeah, it's usually that is called multimodal type of MRI imaging. I mean, the predictions, right? So usually that nowadays people are trying to incorporate the information coming from different modalities. I mean, they have first used the individual metrics, and then they have found some accuracy levels, right? And then finally they have combined this information together, right? They have information together, and they have observed, even my study, we observed there is a increment of this combination of information. That's why actually this paper is getting more value in publication, because usually people do separately, but in our case we have used 3 different techniques, and using these different techniques we got different information. So why do we not combine this information if we are getting 3 different information? That's what our question. So we have tried that, and then finally we got the increased accuracy. So I think even for your research, if you are taking more than one modalities or more than one the indexes, even you know that in structural MRI you can take more than one indexes like SBM, VBM, TBM like information, then you can combine those and you can check for, you're asking whether it is clinical. I think it is very clinically important, because if we can increase the accuracy, means that the classification power, that is the most important thing. That's very important.
Okay, thank you.
Thank you very much.
Okay, if you don't have any more questions, it is time for the time for wind up the session today. So let me thank our guest speaker, Dr. Vasana. On behalf of Radiography and Radiotherapy Alumni Association, I want to express our sincere appreciation to you, Dr. Vasana,
Thank you
for
very much,
sharing.
Dr. Bamali.
Yes, for
Yeah,
sharing.
thank you very much for
valuable.
inviting me. Actually, this was a long-awaited lecture. Actually, I wanted to give something from my knowledge, and hopefully we can work together in future setup or future neuroimaging work. Okay, especially I have a message for undergraduate students. Please do high quality studies, because one of our undergraduate research accepted by OHBM means that it is highly valuable. I mean, that's good for our students as well. Okay.
Yes, of course. I'm pretty sure this session is profitable for everyone in medical imaging. So I would like to thank all the participants today, and looking forward to seeing you soon in the next session. Until then, so good night and stay safe. Thank you very much.
Thank you.